at the back, please. Okay, just to remind you, we've been talking about using antibodies and antibody fragments as therapeutic agents. We started off defining monoclonal antibodies, looking at the structure of antibodies, and then seeing how we can use parts of antibody molecules as therapeutic agents. Sometimes coupled to other molecules, and sometimes we'll find by themselves. This was an example of coupled to another molecule. I'll give you one other example, a variant on this, and that's shown here. And this is a bispecific diabody. In other words, really two single chain Fv fragments. They're coupled together covalently. One binds to a protein on the cell surface. The other binds to some sort of toxin. So again, we have different possibilities. Instead of having the toxin covalently coupled to the antibody or the antibody fragment, the toxin or protoxin can be added at a later time. And we have, again, specificity. We have the toxin directed to specific cells and therefore able to function in a way that leads to fewer side reactions. Okay. I'd like to talk about, and I'll spend a little bit of time on this. Sometimes antibody molecules are too large to be effective therapeutic agents. Sometimes even fragments of antibody molecules are too large. And the reason that it's problematic is because if we're talking about an antibody actually getting into a tumor, it may get into some of the cells on the surface, but it may not penetrate the tumor to any significant extent. And therefore, it's not very effective. And so people argued, they thought, well, if we had a smaller therapeutic molecule that was also targeted, we could get into the tumor and it might be more effective. So the question is how to get a smaller antibody fragment, and by smaller I mean smaller than an Fv fragment. So an Fv fragment is already just about 1/6 the size of a whole antibody molecule. But we want it to be much smaller than that. And what this group of workers started with, they started with, here's an Fv fragment, again, represented conceptually. So I'm showing the variable portion of the heavy chain, the variable portion of the light chain, and I'm emphasizing the framework regions and the CDR regions. And yes, I know that they're not all the same size, but I'm showing them all the same size. And to make it clear that the CDR and the framework regions are different for the two chains, I've used different colors in their representation. What this group of workers did is they said, well, really what we need is we need some framework region and some CDR regions. We're not sure how many framework regions and how many CDR regions, but it seems that we don't need 4 framework and 3 CDR, or we hope we don't. And so they modeled this, and they made from this FV fragment. They made a number of derivatives, and one of the derivatives is shown here. And I've only shown you the one that really worked well, and that is there's CDR1 from the heavy chain, fragment 2, framework region 2 from the heavy chain, and CDR3 from the light chain. Now, why that particular configuration? Basically, I think it has to do with how the molecule folds and the portions of the molecule that can interact with other portions. But in fact, this very small fragment, this very small portion of an FV fragment can bind to the target antigen. And it does it with reasonable avidity. That is, it does it reasonably strongly, so we're not losing much in that regard. And so if it has similar binding specificity, then we can test it. And of course, how do we test it? We do this in, take cells in tissue culture, and we're going to add Epstein-Barr virus. The molecule itself, this short peptide, is going to be coupled to another molecule called a colicin, colicin 1A, and so it is Roman numeral 1, it's not IA. And so the Epstein-Barr virus, first of all, it's tested in cells in culture, but then they tested it in mice. It causes tumors in mice, and when they add this small fragment, and of course they have all the appropriate controls where they add other molecules as well. They add the fragment without the colicin, and they found that this small portion of the FV fragment together with the colicin, which, and that's your toxin if you like, could penetrate the tumor and in fact kill the tumors in mice. Now, is this yet being tested in humans? Not as far as I know. That's logically, after this is done more extensively in mice, after it's tested more thoroughly in mice, logically that would be the next step. Okay, but that's interesting. What that means is, at least for some applications, not for all, for different applications, we're going to need different things, but for some applications, we can get by with a small portion of an antibody molecule. And that small fragment of the antibody molecule in some ways may be better than the whole antibody. Okay. I talked briefly about making monoclonal antibodies and how you have to fuse cells in cell culture and then select them from cell culture and then grow them up. This is a process that the fusion, the selection, the growth takes months. It's not a 5-minute job. It's not E. coli that you're dealing with. It's not simple selections. And that being the case, one would like to speed up that process. Moreover, worse than that, you also have to immunize the animal to begin with and assume that, or hope that the animal makes a significant number of antibodies against the target antigen. And once you've isolated the monoclonal antibody, if you wish to produce a lot of it, what you're left with is growing, and you can remove the DNA from that, you can subclone it, you can produce it in either bacteria or CHO cells, but the scale-up becomes problematic. The scale-up is a big deal. So first of all, the selection is a problem, and the production is a problem. So a lab in California at the Scripps Institute. The Scripps Institute is just, well, I guess San Diego is I guess you can call it San Diego, or just outside of San Diego. And I just want to give you some perspective on the resources that went into doing this work, because the resources were enormous. A typical grant from NSERC, and NSERC funds non-medical research in Canada, is in the neighborhood of $30,000–$35,000 a year. The group that was working on this is done about 15 years ago. This group in California, in addition to the salary budget, so that $30,000 to $35,000 includes salary budget. In addition to the salary budget, there were about 20 or 30 people working on this project. They spent $165,000 on supplies a month. That kind of funding that we're talking about to do this project is absolutely unheard of in Canadian science. Even MRC funding, or CIHR funding, it absolutely, funding at that level doesn't exist. Okay, what did they do besides spend money? What they did is they said, and Richard Lerner is the head of the group, and that's why his picture is there. And I'm going to show you another picture in just a few moments of someone else who took a slightly different approach to this, and that's Greg Winters. And Greg Winters and Richard Lerner are both, they both have somewhat large egos, and they both think that they should win the Nobel Prize for the work that they've done, and they hate each other. And apparently neither cites the other one's work. So just to give you some perspective, while scientists can be generally quite clever and quite nice, it's not to say that they're not very competitive sometimes, in the extreme. Okay, what Lerner and his co-workers did, quite simply, they said, a human being makes so many light chains, so many heavy chains, so many CDR regions, and if we take those and we shuffle them, basically what's happening in the human antibody response and the human immune response, or in any mammalian immune response, you're getting more or less about 1 million different antibodies. And you can get that, if you think about it, from 1,000 light chains mixed with 1,000 heavy chains, and you can get 1 million different combinations. It's not exact, but, and if you think that if you mix up the CDR regions even further than that, you can get much more variation. But you don't need a lot of genes to get the human immune repertoire. And so what Lerner and his co-workers did, they said, let's isolate mRNA from lymphocytes. And they started with lymphocytes of animals, mice in particular, that had been immunized with a particular antigen. And the antigen doesn't matter in this case because we're talking about a principle here. They isolated the cDNA, or they made cDNA rather from the mRNA, and that's easy enough to do. And then they amplified the cDNAs that they had, and they amplified them in two ways. They amplified first separately the heavy chains and then the light chains. And they know enough sequence information on the light and heavy chains because they know they don't know anything about the CDR region sequences, but they know the sequences of the framework regions. So they can amplify the chains. They can then put in specific restriction enzyme sites in those fragments. They can clone them into vectors that they've designed, and I'm going to show you the vectors, and they're really quite simple. And they're using vectors, in this case, that are based on bacteriophage lambda. And you may remember bacteriophage lambda is a bacteriophage that infects E. coli. And it's been used as a vector for many, many years now. And as a bacteriophage vector, typically you can clone pieces of DNA of around 20 kilobases. We're then going to take those inserts, once we've identified them, we're going to cut them out of bacteriophage lambda, and we're going to combine them from two separate bacteriophage into one. But we're going to do it, we're going to have, if you like, we're going to have one pot over here with the light chain variable regions, another with the heavy chain variable regions, and we're going to combine them, and in combining them, we will be producing, if you like, more or less 1 million different antibodies or antibody fragments in bacteriophage. So, and then, of course, we can screen that for what we're looking for, and then scale it up. And part of the scaling up is cutting it out of the bacteriophage and putting it onto a plasmid and then producing it in E. coli. Okay, it all sounds pretty good. It's actually a long process. It was a long process to develop. Okay, let's look at the vectors. The vectors are quite simple, actually. In all cases, the vectors are under the control of the lac promoter. And you'll remember that the lac promoter is an inducible promoter. We typically use, it's not necessarily the wild type lac promoter, it's a mutant of the lac promoter that's a little bit stronger. So first we have A and B just show separate fragments. Here you have a fragment of the light chain, a fragment of the heavy chain. And notice in A, just after the lac promoter, you have an echo R1 site. And then downstream at the end, you have a NOT1 site. And for the heavy chain, you have a NOT1 site right after the promoter, and then an echo R1 site. What that allows you to do, and that makes the assumption, of course, that there's no echo R1 sites in the light or heavy chain of the cDNA. That allows you to splice those two together by digesting with echo R1. So you cut one here, you cut the other one here, and then you can put them together at echo R1. Now they're both under the control of a single promoter, and you have an operon that's making both the fragment of the light chain and a fragment of the heavy chain. And it works. Okay, the interesting, so that's what they did at first, and they took it further. And what they did in taking it further is they said, well, if we have sensitive enough selection, and that's easier said than done sometimes, then really then inoculating an animal is really only a way to enrich for the mRNA that we're interested in the first place, so we can get more cDNA, so it makes this process more effective. But if we're clever enough in our selection, we should be able to go through the whole process without inoculating an animal at all. Just start with its complete repertoire of light and heavy chains, and then select in vitro, really, and come up with the antibody or the antibody fragments that we're interested in. Okay. The other approach to this, I mentioned, is Greg Winter. This is Greg Winter. I guess I don't say here, but Greg Winter is at Cambridge in England, the same place that Milstein and Kohler were working. So they do know one another. And Winter also is going to use a bacteriophage to construct his, and by the way, Lerner called his constructs, when he put these things together, he called it a combinatorial library. And it's combinatorial because it combines two libraries. And so you get all the different possible combinations of those two libraries. Winter is also making a combinatorial library, but Winter's doing it slightly differently. Winter is going to take, and again he's going to start with an inoculated animal. He's going to amplify the light chains and the heavy chains, again separately, the cDNA, that is, and then he's going to splice them together with a linker in between. So he's basically making single chain antibodies, and he's doing this all in a test tube. And he's arguing he doesn't need the step of cloning it into bacteriophage lambda. That's just an amplification step. So he can take this straight from the test tube and insert this into a bacteriophage vector. And the bacteriophage vector, he's going to use bacteriophage M13. And we'll talk about that in just a minute. But he's inserting it basically into the chromosomal DNA of this bacteriophage. Now M13, I don't know if you talked about this in 342 or not, but bacteriophage M13 is an interesting vector. It's called a filamentous bacteriophage. It has a somewhat different lifestyle than lambda. First of all, it has a smaller amount of DNA. Second of all, in its lifetime, it's mostly single-stranded DNA. It's a single-stranded circle, and only during a small portion of its lifetime does that single-stranded circle become double-stranded. That's the so-called replicative phase of the DNA. So the DNA needs to become double-stranded in order for the phage to replicate. And you may remember, and people may not talk about it much anymore, but bacteriophage M13 was used in the Sanger sequencing technique for sequencing DNA. DNA was spliced into the bacteriophage, and when the bacteriophage was double-stranded, it was allowed to amplify, and it was then isolated as a single-stranded version later on. Okay, so what you're doing is you're taking this into, and so here you have your combinatorial library. It's on the bacteriophage genome, and in fact it's inserted into the gene for a protein called gene 3. And you'll see why in a moment. this, as a filamentous bacteriophage, this is not one that has a lytic cycle. It does not lyse E. coli. It may eventually kill E. coli by just producing a lot of its own stuff, but it doesn't specifically lyse it. And it's an elongated bacteriophage. So I've shown it here. These little circles are supposed to be coat protein. Here's the genome, the bacteriophage genome, with the DNA spliced into it. And at one end, we have protein 3. And there's actually 3 copies of protein 3. And so what we have on the surface, the way this is constructed, because we made a fusion protein, okay? On the surface of the fusion protein, we're making a single chain FV fragment. And we're making 3 copies of that. And because any one bacteriophage will contain only one type of single chain FV fragment, we'll get these 3 are identical to one another. But so we need thousands or millions of them to get the entire immunological repertoire. Okay, how do we screen that? That turns out to be easy. And it turns out that this is probably a little bit easier to do, to use Winter's approach. And this is, again, just a cartoon, don't take it too seriously. Nothing here is drawn to scale, okay? This is not a tube or a beaker. This is rather the well of a microtiter plate. So you've seen microtiter plates, they're about that big, more or less. And some of them have 48, some of them 96, some of them close to 200 wells in the plate. So we can do multiple tests on a single plate, and we have so-called plate readers that can scan this. And what we're going to do is, to the microtiter plate, we will bind our target antigen. So if the target antigen is a protein that's supposed to be on the surface of a cell, okay, we have to isolate that and we bind it, and that's shown here in the red squiggly bit. And there'll be lots of it bound, but it'll be to the surface. Not necessarily just the bottom, by the way, but the walls as well. We can automate this whole process, using robots, adding solutions, removing solutions, washing things. And here's our recombinant bacteriophage. And these single chain antibodies, if they recognize this bound antigen, they're going to bind to it. And they're going to bind to it tightly. And I didn't talk about how tightly antibodies bind their target antigen, but they bind very tightly. Association constants of 10 to the 8th, 10 to the 9th, 10 to the 10th. That's much stronger than most enzyme-substrate interactions. So what we can do is we can add our mixture, bind different bacteriophage, and look for the wells that, because now we're going to add, do a secondary test, we're going to add antibodies against the bacteriophage. We're going to look for wells that light up with a colored reaction. We can discard everything else. The fact that we have many wells and we can automate this so we can wash away one reagent after another, we're left with a few colored wells, and from those wells we can isolate the bacteriophage, and then from that bacteriophage we can isolate the DNA and then subclone the DNA onto a plasmid. Again, we're getting around having to inoculate animals, or the possibility we have here, and we're speeding up the process immensely. We're going from months to days in terms of the entire process and the selection. The selection, it turns out, with hybridoma cells is very, very slow. Those cells grow slowly in culture. Okay. I want to say a couple of more things. We also have the possibility, and so this work is more recent. Some of that work was done about 10 to 15 years ago. So in biotechnology, that's getting a little old. More recent than that, somebody said, well, sometimes we want not just an antibody fragment, but we want a whole antibody molecule. And so what they did is they said, okay, we can produce a whole antibody molecule in E. coli, produce light chains and heavy chains, but how do we purify them? How do we get them, not just purify them, how do we select them? And so what they did is they designed an E. coli host that would facilitate this. And so the E. coli host has now a protein, and I've shown this protein, and this is purely cartoon-like. It has a protein that is going to bind and be localized in the inner membrane. That protein is from the E. coli lipoprotein. It's just a portion of it, but that fragment localizes only in the inner membrane. And we're going to fuse that to, and what is it? A fragment of Staphylococcus aureus protein A, or the Staph A protein. Staph A protein is a soluble protein. So we make this fusion, and the Staph A protein is going to be soluble, but in the periplasmic space, in the space between the two membranes. Now what's happened is we have an E. coli host that we're going to use to produce antibodies. The antibodies will be produced. We'll design them so the antibodies will be secreted, or the chains will be secreted. They'll assemble in the periplasm. And the Fc portion, which we've ignored up till now, binds specifically to the Staph A protein. Binds very tight with the Staph A protein. In fact, the binding of the Fc region to the Staph A protein is the basis for a number of immunological assays. It's not a therapeutic thing, but it's a way of measuring. Now, if we take those cells, and again, we look at individual clones of those cells, we can add antigen that binds to our antibodies and also can give us a color reaction. And if you remember, and I'm not showing how you can get a color reaction, but if you remember the ELISA method, where you had a primary antibody, an antigen, a secondary antibody, and then the secondary antibody was coupled to an enzyme, and you can get a colored reaction, and again, easily identifying the clones that you're interested in. The outer membrane, and this all depends on one thing, the outer membrane in gram-negative bacteria is fairly leaky. The inner membrane is quite tight. I've not shown the cell wall here because that just is going to complicate everything to show it. But the outer membrane, it's pretty easy for things to get in, at least to the periplasm. It's also easy for things to leak out from the periplasm. Okay, so we have, again, we have a way of selecting, in this case for whole antibody molecules, not for fragments. Okay, just a couple more things I want to say about antibodies. Suppose we say, okay, in every case are 1 million different antibodies produced by a mammal. Is that sufficient to bind to any molecule that we wish to target? And the answer is sort of, yeah, more or less. And that is, we can get something will bind from the mammalian immunological repertoire, but it may not bind very tightly. If we want something that's going to bind really tightly, be really specific, what we need to do is shuffle some of the DNA, create new antibodies, antibodies that never existed before in nature, and some of those, by the way, most of those shuffled antibodies will be terrible. They won't work at all, or they'll work very poorly, but some of them are going to bind our target very, very tightly. And that's what we're looking for here. So we can, in this case, we're going to start with different antibody coding cells, going to mix them together, we'll extract the mRNA, make the cDNA, and then you have a whole bunch of CDR regions. And even if you start, by the way, with cells from different people, those different people will have, or different animals, they'll all be slightly different. But if we, even if that's not the case, if we amplify the CDR regions, instead of putting them just back where, instead of replacing them one at a time as we did before when we humanized an antibody, what we're going to do is take, and it shows here with the colors, we'll take the different colors, the 6 different CDR regions, and we'll put them together in random ways. So and you can think about, you have 6 times 6 times 6, or 6 to the 6th possibilities of the way these can go in. And so you have a very large number of possible fragments. Again, most of them will give you nothing that's useful. Occasionally you get something that's really very good. And it may be 10 times, it may be 100 times more effective than the antibody that you could have gotten any other way if you limited yourself to natural antibodies. Is this yet being used commercially? No. It's just, so far, it's only been developed at the level of the lab. Okay, one more thing. Sometimes in, let's say, a cancerous cell, you'll see a big difference between the cell surface proteins of the cancer cell and the non-cancer cell. So the cancer cell may make 2 or 3 or 4 new proteins, and they're visible on the surface of the cell. And we can detect them with antibodies, we can detect them in other ways. Some cancers, that's not the case. However, sometimes after we add a chemotherapeutic agent, and that's what we've done here. We're taking the cell, we're adding a chemotherapeutic agent, and all of a sudden, and I'm showing the arrows as sort of bursting out of the cell, all of a sudden we have that cell, and there would be a difference, of course, between the healthy cell and the cancer cell. There has to be for this to work. that cell is all of a sudden now making new cell surface proteins. The new cell surface proteins, now we're going to add an antibody, whether it's a fragment or a whole antibody, and it's going to be conjugated to a toxin. So that's shown here. So the antibody, and again, it's a cartoon, so don't take it too, this doesn't imply too much about mechanism. The antibody is binding to those new proteins on the surface, and it's fused to the toxin. Some of the surface proteins will be translocated inside the cell. The toxin will get inside the cell, and it will kill the cell. Again, it's a way of facilitating killing on targeted cells, typically cancer cells. It's something that's only been developed, again, in the lab at this point. Okay. Now, that's all I want to say about that chapter, but don't go away. Because so far, and in terms of commercialization, most everything that we think that we've spoken about, or most everything that we think about as a therapeutic agent, is a protein. Whether the protein is replacing a human protein that's being defective or missing, whether it's using an antibody to regulate the expression of some proteins, but they're all proteins. That's a lot of work, because it means, generally speaking, we have to make the protein. We have a scale-up issue. We haven't talked about that at all, but that's a lot of biochemistry. And we have to purify the protein on a large scale, maybe a very large scale. And then we have to use that as our therapeutic agent. Suppose we didn't have to do that. Suppose instead we could give not the protein, but some sort of nucleic acid, either RNA or DNA. And we got that to be somehow targeted, taken up, and converted into the protein that we're interested in vivo. We no longer have to do protein purification. We no longer have to do scale-up. All we have to do is make RNA or DNA. That's actually much easier, much simpler, and much less expensive, also less time-consuming. All right, so we're going to talk about using nucleic acids as therapeutic agents. This is fairly new stuff. I mean, people have been trying to do these things for a number of years. The success in this is relatively recent, but there has been some success, and we'll see that. But really, I'm going to talk, I will give some examples of the successes, but really what I'm more interested in here is the attempts, what people are trying to do and how they're trying to do it. So we're going to talk about antisense RNA, and I will define that very shortly. We're going to talk about ribozymes, and ribozymes are RNA that have catalytic activity. We're going to talk about RNAi, interfering RNA, and then we'll talk a little bit about gene therapy, not much. And we'll talk about some of the mechanisms for delivery of these compounds. Okay. When we talk about nucleic acids as therapeutic agents, the major problem, the major limitation in using these It's not identifying the appropriate RNA or DNA. It's not in synthesizing the RNA or DNA. It's delivering it. It's how do we deliver it in small enough amounts, but how do we target it to specific cells? And we're only going to talk about a few methods for delivering, but there are literally hundreds and hundreds of papers talking about different methods for how we deliver these therapeutic agents. First of all, let me define antisense RNA. And what's shown here in A, you can see the figure. You have well, no, we don't want to. Okay, if you look at the right-hand side of A, what you have is you have a promoter and you have a gene. The gene has, in this case, it's shown with 2 exons and 1 intron. The exons are labeled 1 and 2. The intron is labeled A. The promoter is the place where RNA transcription begins or is directed. It directs RNA polymerase to begin transcription. We get a primary transcript that includes the intron, and eventually the intron is removed, it's cut out, as the transcript becomes mRNA. Okay, you're all familiar with that from your molecular biology courses, probably even from high school. There's a single direction, then, with respect to the promoter for a gene. If the promoter is facing one way, the gene has to be facing the same direction downstream from the promoter, and we get an mRNA. Pretty straightforward, okay? What if that gene, instead of facing this way, is turned around as facing the other way? What happens then? We get what's shown in the left part of A, and it's shown in red. You get an antisense RNA. The antisense RNA is the exact complement of the mRNA. So it's transcribed from the other strand. The antisense RNA does not code for protein. Doesn't code for anything. But if you add an antisense RNA, as shown in A, if you add the whole thing to an mRNA, it binds based on base complementarity. One is exactly complementary to the other. So we know that DNA molecules base pair with one another. Well, so do RNA molecules base pair with one another, based on pretty much the same rules. When that happens, RNA forms a stable complex, double-stranded RNA, and translation of that message does not take place. This is shown in B. We have the same thing, just a small variation on this. Instead of having an antisense RNA that's the whole gene, or the whole cDNA, we have an antisense oligonucleotide. So we have a portion. It could be a small portion, it could be a big portion. But we'll just talk about the antisense oligonucleotide. It hybridizes to the message. Again, if it's a strong enough hybridization, if it's a stable construct, it prevents the message from being transcribed. Eventually, the message or the message antisense RNA complex is going to be degraded. So one way of shutting that, if we have too much protein expression because we have some Well, we have aberrant gene expression because of some disease, whether cancer or otherwise. One way, theoretically, of shutting it down is using antisense RNAs to target specific messages. Okay, we'll talk about this more on Monday. See you then.